Chapter 15 The Stuff of Life. In 1861, while the controversies about the cell theory were raging, Max Schultz, the German zoologist, began to examine cells through his microscope. Before long, he had his own statement to make about them. He said a cell was, quote, a mass of protoplasmic provided with a nucleus, unquote. And what was a mass of protoplasm? And what was a nucleus? In about 2 years, he had a pretty good idea of the answers to these questions. And at the same time, he followed his first statement with another, quote, protoplasm is the physical basis of life, he said. Many scientists before Schultz's time had noticed the sap or jelly-like substance that was to be found in animals and plants. Probably Johannes Perzinke of Prague was the first of these. He found it in plants but did not understand what it was. He was a devout Christian and decided to call it protoplasm, taking the name from protoplastus, which in the liturgy of the church was another name for Adam. Hugo von Moll, the German botanist, had seen through his microscope the same substance in the slime-like, granule-filled material that traveled with a circular movement within plant cells. And then Félix Dujardin, studying the single-celled marine creatures called protozoa, noticed it too. He said it was, quote, a substance viscid, translucent, homologous, elastic, and contractile, unquote. He called it sarcode. Okay? But though all these men had seen the jelly-like substance in animals and in plants, none of them attached great importance to it, having perhaps set their eyes on other matters. Then, while the scientific world was arguing about the cells that Schleiden and Schwann had seen, Max Schultz asked his question and gave his answer. At first, little attention was paid to him, but gradually the biologists began to realize the importance of what he had said. By the time the 20th century had come, they believed that the study of the cell with its protoplasm and nucleus might give them the key to understanding life itself. It is a pity that more has not been written about Max Schultz. We know that his life was short. He died at 49. We know that he was a lecturer at the University of Halle in Prussia and a professor at the University of Bonn. We know that he loved music. He kept his violin close beside his microscope. And we have his picture, the picture of an intellectual, sensitive man wearing a dark suit and a bow tie and with a straggly beard. Perhaps he would prefer that the details of his personal life not be recorded. Certainly he would feel that his work was more important, and there is plenty of information about this. In his laboratory at the University of Bonn, he was known as a hard worker, particularly when it came to his microscope. Soon he was famous for his studies of amoebas, the little single-celled animals that look like tiny globules of jelly moving through seawater. He studied the ramifications of the nervous system in vertebrates too, and made important contributions to the knowledge of electricity in animal bodies, but his great contributions grew from his study of the amoeba. For he realized that these little globules of jelly-like stuff were in fact living creatures. They had the power to move from one place to another, changing their shapes to form little false foots, and then, after they had moved, draw them back again. When he touched them or passed a slight current of electricity through them, they changed their form. He saw, moreover, that they seemed to consume nourishment and excrete waste, and that they took in oxygen and gave out carbon dioxide. And since they also had the power to reproduce by dividing themselves into two parts, he knew that they were animals. Looking down through his lens, Max Schultz saw the stuff of which the amoeba was made. It was a mass of jelly containing a nucleus. The jelly sometimes grew cloudy, sometimes was clear. there was movement in it. Sometimes it seemed to be a whirling movement, sometimes a hardly perceptible drift. Others had seen this movement before him. Robert Brown had seen it first, and so scientists call it the Brownian movement. They did not know what it was. Max Schultz began to examine the jelly-like substance in other animals and in plants. In animals, the movement seemed to be rather slow, but in plants it went whirling around at a great rate. Strange, he must have thought, for we have thought of plants as stationary and animals as moving. He could not understand it, but he knew that the movement in the protoplasm was the movement of life. When the cells died, the movement stopped. In 1861, he was ready to publish his findings in a short and rather unassuming essay, which he sent to the German Journal of Anatomy and Physiology. In the beginning of this essay, he made the statement that a cell was a mass of protoplasm containing a nucleus, and he went on to say that it would be impossible for a cell to have surrounding walls. If it had walls, how could it divide in two? And if it could not divide in two, how could it reproduce? It would be like an imprisoned animal. It ought not to be called a cell at all, he reasoned, for it was almost impossible to think of a cell without walls. The cell of a honeycomb has walls of wax. A monk's cell or a prison cell, both are spaces surrounded by walls. Still, since biologists had spoken of cells for so long, he did not suggest altering the name. Yet he declared the cells of which all living things are made are simply infinitesimal masses of jelly-like protoplasm, each with its nucleus. Of course, he said, they have no walls. Moreover, he said, the protoplasm that surrounds the nuclei in different tissues differs not because of any substance foreign to the cell. Its different forms are simply a transformation of the protoplasm itself. The very name protoplasm, which formerly had been used only by botanists, should be the universal term for the substance that is fundamental to life. It is found in plants, in lower animals, and in higher animals. Its consistency is different in different animals and kinds of plants. Sometimes a number of nuclei can be surrounded by this protoplasm, which can afterwards separate and form separate cells. That was the substance of the essay Max Schultz published. Since then, microscopes have been greatly improved. Now they can magnify up to 400 and 500 times and more. And biologists have found out more about the protoplasm that is the stuff of life. Protoplasm is a substance which scientists call a colloid. It resembles glue or white of egg. More than 80% of it is water, and in this water float tiny particles or granules which combine and recombine, break apart, and drift together again. It lives by change. At times, the protoplasm appears perfectly clear under a high-powered microscope. At other times, it is grayish again, and there are tiny particles floating in it. Most of the changes cannot be seen even through the sharpest of lenses, but as the tiny particles shift, join into new combinations, and separate again, the whole colloidal system changes, producing movement, growth, and other vital functions. Living, therefore, is the sum of all the changes that take place in the protoplasm. As they watch the changes and the movements in the living cells, scientists have sought to find out what protoplasm is made of. Can it be made of the materials that are found on Earth, or is there some special essence, some peculiar force in it that does not belong to the material of Earth? Does protoplasm have any elements in it that are not already on Earth? Here chemists have come to the aid of biologists in an important new branch of science known as biochemistry. What? Though their study is still in its beginnings, this is what they have found. Every kind of protoplasm contains 9 elements. These are oxygen, carbon, hydrogen, nitrogen, potassium, phosphorus, sulfur, magnesium, and iron. All these are plentiful on the surface of the Earth. Some kinds of protoplasm contain additional elements. Calcium, sodium, chlorine, and copper have been found in them. But these are common Earth elements too. Therefore, up to the present time, the biochemists have found nothing to make them believe that protoplasm contains any substance that is not contained in water, air, and the rocks of the earth. How these material substances combine, why they react as they do, whether it would ever be possible for scientists to combine them and to create life, these are unsolved questions. But even as we ask them, the wonder and the mystery of life remain. What is it? What is this protoplasm, this elemental jelly that Max Schultz found in all living things? What is the stuff of the oak tree digging its roots into the ground, the daisy blowing in the wind, and the boy running across the field? They are mysteries now as they were before high-powered microscopes were ever invented. Thomas Huxley, the 19th century English scientist, made a singular description of protoplasm as seen through a microscope. Incidentally, he had been a surgeon on the British ship HMS Rattlesnake, where he had made a serious study of the surface life of the tropical seas. Concerning his findings, he sent back, quote, communication after communication to the Linnean Society, unquote. When The Origin of Species was published, he found in it a good working hypothesis for evolution. Having examined the cell of a plant through a microscope, Huxley wrote, quote, a spectacle afforded by the wonderful energies imprisoned within the compass of the microscope tell of a plant which we commonly regard as a merely passive organism, is not easily forgotten by one who has watched its movement hour after hour without pause or sign of weakening. The possible complexity of many of other organisms, seemingly as simple as the protoplasm of the plant just mentioned, dawns on one, and the comparison of such activity to that of the higher animals loses much of its startling character. Currents similar to these have been observed in a great multitude of different plants, and it is uniformly believed that they occur in more or less perfection in all young vegetable cells. If such be the case, the wonderful noonday silence of a tropical forest is due, after all, only to the dullness of our hearing, and could our ears catch the murmur of these tiny maelstroms as they whirl in the innumerable myriads of living cells that constitute each tree, we should be stunned as with the roar of a great city.